Christopher Cousins' research is focused on torrent salamanders, a family of salamanders that's endemic to the Pacific Northwest. Using multiple genomic and modeling tools, Chris's research is at the forefront of efforts to save these sensitive and elusive creatures. He's developed tools for using environmental DNA to take water samples from streams, use DNA to determine if torrent salamanders are there. He's currently doing fieldwork to use landscape genomics to understand what landscape features act as barriers or facilitators to the torrent salamanders' movement and reproduction. In addition to his PhD research, Christopher is also a co-author of a bilingual children's book about environmental stewardship called Nuestra Casa. More info can be found at www.nuestrabooks.com. I'll put that in the chat as well. Welcome, Chris. Take it away.
Hi, thanks. Okay, let me do this. Okay. And give me 1 sec, sorry. Give me 1 sec.
No problem.
Okay. Okay, is that working fine? Okay, great, thanks. Well, thanks for the introduction and thanks for having me. So yeah, it's fun, always fun to talk to a different crowd. So this is be a little different maybe than some of the presentations you've had. So I just kind of wanted to give you a roadmap of kind of what the presentation would be and kind of where we'll go with it. Because there's a lot. I tried to kind of fit in a lot so you can kind of get an understanding of what I do and how it relates to conservation science. So we'll start with kind of a broad overview to headwater streams and the amphibians that are found there. Then kind of move into torrent salamanders, which is the taxa that I worked with, which, you know, as was mentioned, are endemic family just found in the Pacific Northwest. And then we'll kind of move into more of the conservation science. So this is where my thesis really tries to answer questions and provide data to the actual managers of Fish and Wildlife Service and people that are able to use that data to make the best informed conservation decisions that they can. And then I'll kind of end by talking about some of the outreach that was mentioned, the children's book that I was lucky to be a part of. So we'll kind of jump into talking about headwater streams. So, you know, this is a term that gets tossed around a lot, but essentially what it refers to are the streams that are the source of all downstream water. So kind of in this diagram, like any of the streams that are labeled with the number 1 are going to be headwater streams. So going all the way down, feeding into more and more streams until you get like a number 4, which would be kind of like a Willamette River. And as you can kind of imagine from looking at the diagram, these headwater streams actually comprise the bulk of all stream networks. So they're the source of all downstream water and consist of about 80% of all streams worldwide. And that actually is also the case in the United States. And so, of course, they have like a lot of hydrological importance, but they're also very ecologically important. So as you can, again, kind of tell by looking at the diagram, these are much smaller streams than some of the downstream. And so what that means is they're much more closely tied to the riparian or stream side forest around them. So you get a lot more interactions between wildlife and plants that live on the forest and then with those that live in the headwater streams, as opposed to something like with Willamette, where you get much more of like an aquatic and then a terrestrial species groups. You get much more mixing in headwater streams. They're also home to pretty unique communities that are found kind of nowhere else. There's a lot of species that are really physiologically developed for this habitat and not for more of the downstream reaches. And it's thought that they contribute disproportionately in biodiversity. So they're home to a lot more of the species richness in these forests than more of the downstream. But despite this, they often get a little less protection than other downstream reaches. And that's because headwater streams are largely fishless. So the dominant predators are things like salamanders or macroinvertebrates. And because of that, they're not afforded kind of the same protection that fish-bearing streams are, where, for example, with logging, you have to leave like a certain amount of trees on either side of the stream to protect it so that the fish spawning habitat is still there. Those protections aren't then afforded to fishless streams. So often that can kind of have a negative impact on headwater streams. And I just kind of wanted to show, this is a really good graphic representation of just kind of what makes headwater stream systems so unique. So you can see headwaters are on the far left in that zone 1, and just see how steep they are compared to more of the downstream reaches, again, how small they are. So these are very fast flowing, oxygen rich and very cold streams, as opposed to something downstream in, say, the zone 2 or 3. And they can look very different from one to the next. So these are all headwater streams in the Pacific Northwest, in the Coast Range, and then on the western side of the Cascades, where I do a lot of my research. And you can see just how different one can be from the other. So like on the far left, you have this essentially like a bedrock slide, very steep, and there's not really any substrate, and the plant life that's there is actually more kind of mossy or using those moss mats as a substrate. Whereas then kind of over to the right, you get a little bit more of that incised channel and kind of more of a forested landscape, but still a pretty small stream, and then kind of moving into something with a little more large gravel substrate, more of the kind of that fern stuff that you see in more kind of mature forests. And then kind of even some of these bigger streams, well, that stream looks pretty big. That's actually still a headwater stream, as there's no permanent tributaries that feed into it. But then you get into a lot more of like the cobble landscape. So you can get a lot of diversity in what a headwater stream is, and not just from one stream to the next, but even within the same reach of a stream, things can look quite different. And this is, these photos kind of highlight another kind of interesting attribute about headwater streams, and that's that they're often intermittent. And so what that means is there's kind of surface flow that's going on throughout the stream, but you have large sections of the stream which don't have any surface flow. The flow is restricted to subsurface. So like on the left, this is at a site near Wyeth, if y'all maybe know where that is, in the Columbia River Gorge. It was part of the burn there. And so it looks very dry, as you can see, but there is water above this point at which we actually sample torrents. And there's water below this point as well. And so headwater streams often have that kind of interesting feature, and a lot of the animals that live there have to then be adapted in order to deal with kind of that loss of discontinuous water flow on the surface. And so in the Pacific Northwest, I work with amphibians, and amphibians that live in these headwater environments generally fall into one of these kind of 3 groupings. So we have kind of the obligate red herring and breeders. So this is something like a Torrent salamander, where it doesn't actually use the water in the stream itself, and instead it kind of depends on the stream effect of the kind of cool, moist conditions that permeate upslope, again, in these kind of stream systems where the stream is very closely tied to the forest. And so both the adults and the eggs are really susceptible to desiccation. And so being within that stream environment makes it so that they're able to stay moist year round. Then you have something like a red-legged frog, which is much more of a facultative stream breeder. And so they do use the streams to breed, but they can also use other habitats either for breeding or different life cycle processes. For example, you know, the red-legged frogs often breed in more pond or wetland environments, but end up using headwater streams a lot during the summer to feed. And so, you know, something like this does use headwaters, but it also uses other environments. They can be pretty tricky for conservation, as in order to protect, say, a red-legged frog, you need to protect both the breeding habitat and then this kind of other habitat that's used for different parts of the life cycle. And then finally, you have obligate stream breeders like this Torrent salamander. So these are dependent on headwater streams for both reproduction and larval rearing. And they have some really interesting kind of physiological adaptations to these cold, fast-flowing waters. So kind of to talk about those adaptations, here on the left, you can see a tiger salamander. So it's, as you can imagine, probably closely related to the axolotl, and it has those kind of big floofy gills that we all know and love. And the reason it has those big floofy gills is because it kind of lives in lake or kind of slow, kind of almost stagnant water. And so there's not a lot of movement. And so they're using their gills to breathe and draw oxygen from the water. And the way that they do that is maximizing surface area. So they have a ton of filaments and just these big fans of gills where essentially, you know, the blood pulls in oxygen from the water. And then comparing that to something like a Torrent salamander larva here in the middle, you can't even really see the gills in the photo. You actually have to zoom in quite a bit. And so they are the same structures as the tiger salamanders. You can see these really thin gills, and you can still see the red and the blood. But these are living in really fast flowing cold water where just oxygen is kind of always rushing through them. And so they're able to live with just these small gills. However, if the stream were to, say, get more dry or the water flow was to cease, then they would be in trouble because they don't have the ability to kind of capture oxygen passively like these tiger salamanders do. So kind of talk about torrent salamanders. So yes, this is I think a pretty unique taxa. This is a family that's only found in the Pacific Northwest. They're millions of years old. They've been here for a long, long time. And they consist of 4 species, all in one genus. So there's the Olympic torrent, which lives only in the Olympic Peninsula, the Columbia torrent, which is just south of the Olympic on the Coast Range until about Lincoln City in Oregon, the Southern torrent, which is south of that and up until about Northern California. There's kind of an interesting disjunct population which occurs on the North Umpqua and then kind of south of the Mackenzie. And then you have the Cascade torrent salamander, which is on the western slope of the Cascades from about Rainier to West Fur. And those 4 species are all that's currently recognized. And so again, they live in these headwater streams, but they also live in these seeps and hyperrheic zone, which I think is one of the coolest thing about them. So the hyperrheic zone is something that often, like even in my field, is really talked about that much unless you do stream work specifically, but it refers to this subsurface part of the river where you get some mixing with the subsurface river flow and then groundwater. So kind of in this diagram on the left, you'll see that the water is kind of coming out of the stream and then going back into it, but it's moving under the surface. And kind of the diagram on the right kind of shows how that can mix. You know, you get movement from the hyperrheic zone in and out of the stream, and then you can get movement with the groundwater as well. And this is really cool. And the reason that they're able to kind of live in there is because they've needed to do that to adapt. You know, as you've seen kind of some of the intermittent streams where there's just not a lot of like surface water. There's certain points where they have to move under the surface in order to exist, which is really, really cool. And one of the reasons they need to do that is because they have a very long development time. So this is a torrent salamander kind of near Larch Mountain with some eggs. This is really cool. It's only the 2nd known observation of eggs for this species, and a friend of mine found them and was very kind and let me know where they were. And so we went and checked it out and wrote about it so that cute little note that should be published soon. But the long development time of these eggs is wild. It's up to a year, where normally this is kind of measured in a couple months. And the larva can take up to 6 years to fully transform into adults. And so what that means is for, you know, many, many years, they're reliant on having this kind of cold, fast flowing water. And it can be subsurface, but it does need to be there because the larva will like dry out or just not be able to breathe and won't be able to move to another area. And even the adults, when they're transformed, are still sensitive to desiccation or drying out, even when compared to kind of other salamander species. They have a very low temperature threshold. I mean, streams where the water gets above 8°C, it's really, really uncommon to find them, which is pretty low temperature. And so, as you can imagine, with kind of these very specific needs, climate change and other threats are predicted to impact torrent salamanders pretty heavily. So logging is perhaps a little bit more of an issue for some species like the Columbia torrent, where a large part of their range lies within public land, and maybe not for something like the Olympic torrent, where that's a different land ownership. But what logging can do is not just remove the trees, but when the trees are removed, then the stream is subject to more solar radiation from more increased sunlight, so it can raise temperature and reduce the oxygen that's in the stream, and it can also kind of add more sediment as well. And they need kind of that kind of gravelly substrate to be able to move into the hyperrheic zone. So when you get stuff like silt, it kind of closes that interstitial space, or that space between all of the particles, so they're not able to move through it. Fire, again, is very similar to logging, although, of course, you get the increased stream temperatures immediately. And then with climate change, there's predicted to be both warmer streams and then reduced stream flow. So this is a graph from a scientist on my committee, Dr. Didi Olson and Julia Burton from 2019, just kind of showing each bar graph showing a different climate scenario and just showing how over the decades, we're predicted to see kind of an increase in the dry length of these headwater streams. And that could be problematic, as we don't know how much of that dry length is going to have hyperrheic flow or how much is not. And even from, I never thought I'd be kind of studying this kind of like hyperrheic hydrology type stuff, but I'm kind of having to as part of this species And even the people that, do it as part of their research talk about how difficult it is to truly map this kind of hyperrheic flow that's going under the surface. And so there's just a lot that we don't know. So kind of as a result of those threats, two of the species are currently being considered for candidate species, the Cascade and the Columbia, for listing under the Federal Endangered Species Act. And my thesis really tries to kind of look at conservation science and meet some of the goals. So the goal of conservation science, of course, being to just protect and conserve these threatened species. And in order to do that, we need data. And kind of that can fall into kind of these three sections for my thesis. The first just being, where are they on the landscape? We can't protect what we don't know is there. And with a species like a Torrent salamander, it can be very difficult to find with traditional surveys. We talked about how they can live in this hyperrheic zone and this underground zone. And so we need additional kind of novel tools that are able to detect them with a high probability, even where we're not able to do that physically. And so that's where the first chapter of my thesis came in, this environmental DNA, or eDNA, which is a really cool technology. And in the last 2 decades, it's moved from being just kind of this novel, new method that people were doing research in to really being used heavily as a management tool. And so what environmental DNA does is it essentially looks for passive DNA that's just present in the environment. So like my dog's hair that's on my clothes would essentially be environmental DNA. It's just there. I don't have to collect it from the organism itself. I can take it out and sample it. And in aquatic systems, for example, you have something like this shark, where the shark is constantly, well, maybe not, hopefully not constantly, but it's defecating into the water. It's shedding scales. It's losing part of its skin as it rubs up against things. And all of this results in just kind of this tissue that's floating throughout the water. And so when you collect water samples, you're hoping to capture some of that tissue so that you can then look at the DNA. And that can be really, really useful, especially when you're looking at species that can be hard to find. And kind of from these water samples, you wouldn't be just getting the shark, but kind of all the fish that move through there. So it's a really cool and powerful technique. But there's kind of a lot of caveats / complications with it. So for example, like in this figure, this is a stickleback, and as it's releasing this DNA and as it's being kind of just released into the environment, there's a lot of different things that are hitting it. So light can break down DNA, temperature can impact how fast DNA degrades. There's microbes and enzymes in the water which are going to be breaking it down, and just how long it's been since the DNA has been shed is going to have a big effect on how much is present. And kind of looking at this graph, which I absolutely won't go into all the details of, from 2016, there's a lot of just on something about the origin of where it came from. Was it coming from the individual itself, or maybe a predator was eating it and some tissue flew off? Was it scavenged? Was the state of the DNA? So like, was it in a cell? Was it out of a cell? Is it pieces? Is it a whole strand? How is it moving through the environment? Is it moving through? Is it going under rocks? Is it getting stuck to the spaces on the side of, say, sand particles? And then how is it degrading over time? And then all of these factors then have to be considered when you're thinking technically, like, how much am I going to sample? Where am I going to sample? And all these things. So it can be kind of tricky to get right. And so it hasn't been used for a lot of taxa. Essentially, the way eDNA works is that you kind of collect these water samples, like I said, but then you need to pull the DNA out of it. So in order to do that, you run it through a filter, and kind of that filter then contains the DNA. And there's been a ton of DNA studies, and like I said, it's being used as a management tool now. But a lot of that is focused primarily on fish, and though it has been used for some amphibians, and it's actually used for a lot for invasive plants. That's one of the ways that they're able to detect if it's in a system before it's too big to control. We weren't really sure if it would work for Torrent salamanders, specifically because most of the studies have been on things that live in the water column. But like this salamander here, they often are in substrate. Even when we do find them in streams, because we don't dig down to the hyperrheic zone, we find them usually under a lot of objects. And so it's unclear how much of that DNA is really kind of getting out and how much is getting stuck to the sediment around it. And this photo just kind of like highlights some of the challenges that we thought we might face. So when we sample water for eDNA, we're sampling from the fastest segment of the stream, the thalweg. But that's usually not where Torrent salamanders are. So in this site, which we use for both eDNA and some of the later work I'll be talking about, we only found the salamanders at this one site, which is hard to say that it's even the seep from this picture, but it's this kind of small seep that feeds into the larger stream. And so in order to detect them with eDNA, assuming that's just where they were, that DNA will need to have traveled down this seep and then back into the main channel. And we should be able to like hopefully pick that up. But it was a bit unclear if we would be able to do that. So we ended up doing part of a study we ended up doing a study as part of a larger study that Dr. Lindsay Thurman, one of my committee members, kind of spearheaded for both the Columbia and the Cascade, the two ESA candidate species, where we sampled 98 streams in order to provide data for her project that was really focused on looking at how habitat variables impact suitability of the habitat for torrents. But we took a subset of those where we knew that they were present. So 12 for each species. And then we did these paired physical and eDNA surveys to figure out if eDNA would work. And so for physical surveys, we'd spend 2 hours, either Mitch Ralston or Connor Dooley and I would spend 2 hours total search time per site and then record individuals and some details of the site. And then we would pair that with 12 2-liter water samples per stream, which we then use this peristaltic pump to run through and store in the filter. Then we take those filters back to the lab, extract the DNA, put them on these qPCR plates, where essentially every sample would be divided into 3 replicates. And so we'd run a reaction for each sample 3 times, and then run it through a qPCR machine. And that just stands for quantitative PCR. So it's a way to do PCR that you can essentially look for single species. So we developed the methods, and if you would like to hear more about that, we can certainly talk about it afterwards. And we developed the primers and probes for that. But yeah, just ran these extractions, sequenced it, and tried to see if we could find them or not. So to kind of run through the results a little bit, I just want to explain the figures. So for every site, there will be a circle, but at every stream, we took 12 water samples. So I've broken that circle into these kind of 12 slices. So each one of those slices represents a water sample. So if it's blank, we didn't detect it. And if it's filled in, we did. But then again, each sample was also then broken up into 3 qPCR replicates because we ran that reaction 3 times for each sample. And this is pretty standard for eDNA and most genetic work when you're doing stuff like this, because within even your sample, it's unclear how spread out that DNA is. If you give a really high concentration of DNA, all of these reactions are going to find it. But if there's a small concentration, which is expected for eDNA because it's kind of this passive free-floating stuff, you would expect to need a bit more replicates. So jump to the results. This is what our surveys look like for Columbia torrent salamanders. Pretty cool. We detected them in 13 to 13 sites, which is fantastic. 90% of the samples we were able to detect them in, and 75% of qPCR replicates, which show that the technique works quite well for Columbia torrents. There is kind of that one site, Edward, over here, which is kind of on the left in the center, which is interesting because we didn't find them a lot there, but that was a site where we actually only found them at a seep that was about 50 meters upstream of where we sampled. So the fact that we were able to detect them at all is pretty extraordinary. And then when we look at Cascade torrent salamanders, we did still find them, but the results are a little more mixed. So we found only 10 of 12 sites. And as you can see, there's a lot more kind of variability in what we were able to find. Only about half of the samples amplified, about a third of the qPCR replicates. And that's even more kind of stark when you compare the two side by side. And so it does work for both species, but it clearly works better for Kizari. However, we think that could possibly be just based on a lot of higher abundance. We found a lot more Columbia torrents than we did Cascade torrents. But we wanted to kind of ask more specific questions to kind of better inform people that use these tools. First being just how many samples do you need? And then, were the detection probabilities that we saw correlated with the observed abundances that we had? And then, do stream substrates impact detection probability? And so in order to do that, we use Bayesian hierarchical models. All that, I guess, the big takeaway from that is that we looked at detection probability at 3 distinct levels. So the first just being what was the probability of detection at a site? And again, we knew that they were present at all of these sites. The second is knowing that it's detectable at the site. What's the probability it's detected in the sample? And that's just kind of how much DNA is pouring through the water at any given moment that we're able to find it within a sample. And then finally, at those 3 replicates per sample, what was the detection probability, knowing that it's in the sample? And that's kind of more of a question of not how much DNA is in the water, but as the DNA is moving through the water, how concentrated in it, how much is getting captured in each sample. So we kind of looked first to kind of see, well, how many samples do we need to be able to detect it? And we found that there was no decline when we went from 12 to 6 to even to 2 samples at any of those 3 levels, which is pretty cool because that suggests we can get away with maybe sampling in a little less intensity. And then we wanted to look at that question of observed abundance and eDNA detection probability. So on the x-axis here, it's the number of salamanders that we found at a stream. And then on the y-axis is detection probability. So you can see this very clear relationship where as we find more salamanders in the stream, we're actually getting higher DNA probabilities at the PCR replicate, which is really cool. Number one, just because it kind of shows that our method works pretty well, but also that maybe we were doing a good job finding them. Because if the 2 are correlated, then we're probably seeing that how much we're finding by physically serving is how much DNA is in a sample at any given point. So it made us feel good for 2 reasons. And then finally, like, how is the substrate related to that? Like if you have a stream with silt, is it going to be the same detection probability as a stream that, say, is all boulders? So on the x-axis, we have the medium diameter of substrate. So essentially, at the left of the x-axis, we have stuff like silt, and then you kind of move into sand, gravel, cobble, boulder. So as you move to the right, it's getting bigger. And we did see that kind of streams that had larger substrate had higher detection probabilities for Columbia torrents, but not so for Cascadian. We're not really sure what that could be, though we did have pretty low abundances for Cascadian. So finally, kind of in summary of the eDNA stuff, the method works, and potentially with lower sampling effort, which is awesome. This is Connor, and I'll just say these are very steep stream basins, and so carrying all that water out can be a lot, and so it'd be nice for all of us to be able to only carry 2 2-liter samples as opposed to 12. We did see that abundances were correlated to detection probability at the PCR replicate level in the site, which is pretty cool. And then we did see a possible relationship between substrate and detection probability, but we need to explore that further. So that kind of answers that first question that managers would need in terms of giving them a tool to find out where they are. But we also need to know how they're doing. And the way that we chose to answer that is using genetic diversity. So genetic diversity, of course, is the basis for all biodiversity. You have genetic diversity at the individual level where, you have DNA, we're talking nuclear because that's what we ended up choosing, where you have nuclear DNA from both parents. So you can be, say, homozygous, where you have both copies of the same allele from each parent, or heterozygous, where each parent gives you a different allele. So you have genetic diversity at that individual level, but then that carries into the population level, where some populations have larger diversity than others, and then even carries through into the species level. And genetic diversity matters a lot. It's very closely tied to higher adaptive capacity and a bigger potential for long-term survival. And what you can kind of think about with that is just looking at this kind of population on the left that has a very diverse genetic bank. If, say, a disease is going to happen or kind of a shift in environmental conditions, there's a larger chance that an individual in that population is going to possess the genetic raw material in order to be better protected against that stressor. But with kind of a population on the right where it's much more small fragments of populations, there's a higher likelihood. And then, if, say, one of these 3 populations had it and the other didn't, because there's not really gene flow and the diversity is so low, you might get extra patients, whereas you'd see up to an entire local population die. And so that genetic diversity can be really telling to just know how things are. And there hasn't been any of that done at a range-wide level for Cascade torrents. And that is interesting because they are fragmented. So this kind of shows the range of Cascade torrents in each box. As it gets from light purple to dark purple, is going from less known sites to more known sites. And you can see that there's, the most known sites are the Columbia River Gorge. And it's not a huge surprise as it, is attractive to more than just people. It's perfect for Cascade torrent salamanders. So it's a real hot spot. But as you get farther away from that, you get less. And you see that just the patchiness. There are sites were kind of large areas in the landscape where the habitat should be suitable, but yet they're not there. So there's likely some fragmentation going on, which isn't a huge surprise given how topographically complex the west side of the Cascades is. And there's some big barriers like the Columbia River. There's a lot of volcanic activity. So there's a lot of reasons that we would expect fragmentation. And even within the range of this one species on just the Western Cascades, there's a lot of variability in habitat. So that variability could be reflected by some local adaptation. But again, there's only been that one study from 1987, which when it initially split one species into 4, and it only included 7 sites for Cascade. So we wanted to increase that and sample some unsampled sites to try to understand where these kind of distinct and unique populations are in the landscape. So last year, we studied, with the USGS, sampled 144 individuals across 27 sites, 57 in Washington, 87 in Oregon, and we took about 1 centimeter of the tip of the tail from torrent salamanders in the field and stored that in ethanol, so not the whole animal. And I do kind of want to know, we had 0 mortalities during this, and often we would find individuals that we had sampled later. And reading through some literature when we were kind of developing these methods, we saw that some people were doing kind of toe amputations way back in what I would call the cowboy science days, and they found that these toes would be regrown within a month of larval salamanders, which suggests that torrent salamanders have a really high ability for regeneration. So we felt pretty good about kind of the ethics of this method and that we weren't harming the salamander, but we were able to get this really important genetic material. So once we had that genetic material, again, this is a section that we can talk about it later if you have questions. But essentially what we did was took this DNA, threw it in a reaction with some restriction enzymes where the entire genome would get cut, and then these kind of SNPs would be found. So a single nucleotide polymorphism where just one base pair is different. And we assembled kind of a big bank of that. And we had to do a lot of things because kind of a fun side thing to know is that the genomes of salamanders are orders of magnitude higher than other vertebrates. For example, ours is about 3 gigabases, and torrents are thought to be between 30 and 50, and there are some salamanders that have like 120. We don't know why it is, but they have very big genomes. So you kind of need methods like this in order to have enough variability to analyze. So jumping into the results, this is a PCA plot. So what essentially this shows is that each site, kind of numbered, has had all of the variability from one to the next, taken and then reduced into these 2 principal components. So PC1 on the x-axis and PCY on the y-axis. And these are essentially the components that describe most of that difference. And 50% for the combined kind of explanation is quite high for a PCA, so we feel pretty good about it. And what immediately, of course, jumps out to you is these 4 sites are wildly divergent from the rest of the species. And that's a level of divergence that isn't normal within a species. And so we're a bit concerned, maybe it's kind of one of the other species that has been mislabeled, or there's something else going on. So in order to kind of look at Cascadia itself, or what we're calling the core population, we remove these 4 sites, as this kind of level of differentiation is going to be hiding a lot of the differentiation within the rest of the sites. So once we did that, we have something that looks more of what we expected, where we have this Southern Oregon clade, which is very tightly linked to itself, this part here, which is Mount Hood, and then the western side of the gorge, which is closely related to, and then another cluster that contains the eastern side of the Columbia River Gorge, both above the Columbia River in Washington and below in Oregon, and then the rest of Washington. And that's not what we were expecting. We were expecting that the Columbia River would just have cut these 2 populations in half. So we wanted to kind of take a closer look. So that was kind of a PC where it's not a specific like model framework. So we used a model framework, fast structure, where we were testing kind of these different values of K, or how many unique populations are in the landscape. And using a model allows us to compare each K and have a measure to say which is more likely given the data. So we tested these values from 1 to 8 with a large data set of around 40,000 SNPs. Though from going to kind of smaller to big data sets, they have all given roughly the same answer. So we also feel good about the robustness of that. So once we identified 5 as the best fit for that model, that 5 populations, we then mapped it. So here you can see these 5 populations, which again correlates to the ones that I told you about and showed you on the PCA. But what really jumps out is interesting is that there's only 2 populations that seem to be mixing. And they're right here, again, over the Columbia River Gorge, which is really wild. It's not what we would expect to see. You have this western, southwestern, which is green on the Oregon side, and then this yellow, which is on the eastern side of Oregon, south of the Columbia River Gorge, but it's mixing with stuff across the river. And there's even, it's hard to see in the photo, but the slightest slimmer of that western in one of those populations as well, which suggests that there is the gene flow there, and actually more recently than anywhere else on the landscape, which is a bit wild. And so that in the middle there is the Bridge of the Gods by Cascade Locks. And a friend of mine kind of joked, like, are they crossing over it like at nighttime? And I think that's really cute. And I would love to imagine it. But of course, that's a bit silly. But Bridge of the Gods was named after traditional ecological knowledge from the Klickitat tribe. And so when the settlers came, the Klickitat told them how their ancestor crossed from Oregon to Washington. They would go back and forth and visit their friends on the Bridge of the Gods. And of course, like, Westerners didn't believe this. And we now know that that actually was a real thing. The Bridge of the Gods was the Bonneville landslide, which connected Oregon and Washington and completely cut the Columbia River. And this happened about 1,000 years ago. And most of the Columbia River Gorge is actually highly geologically active. So this was pretty unique. I did not know about this prior to this study and only learned about it because of it. And when we kind of take a zoomed in look and overlay that Bridge of the Gods, we see that it's smack dab in the middle of these populations, which seem to be breeding, which does suggest that they were able to use this bridge to connect. And we do know that kind of the upstream part of the Columbia River Gorge actually ended up forming a large lake. So it's likely that the populations where we sampled were wiped out when that landslide happened and then had to be resettled. So that could be possibly why we see this connection. So kind of in summary, we saw evidence for 5 distinct populations, evidence for a pretty recent gene flow event across the Columbia River Gorge, and some populations are wildly divergent. So, and we're still trying to figure out more kind of the questions of how diverse is one population to the next. So then kind of that last part of conservation science as well, like how do they move across the landscape? And that's where we use landscape genomics. We do kind of the same markers that we had for the population stuff, but now we kind of overlay landscape features like distance and fragmentation to try to understand how they disperse. These salamanders are too small for GPS backpacks. And so this is kind of the only tool we really have to be able to look and see how they're dispersing across the landscape. We're all very jealous of deer and bird biologists. And so what we can do with that data is kind of test some different theories. So, is what we're seeing in the landscape a bunch of isolated populations that maybe have very little gene flow? Is there gene flow, but it's really only terrestrially, or maybe it's only aquatically, like they're moving along streams? Perhaps it's only going within canyons, you're not seeing a lot in higher elevations. Or is it just kind of this isolation by distance pattern where just as you get farther away from one population, it gets kind of, say, more diverged, but in a predictable pattern, so the landscape isn't really affecting that. Or, and this is extremely unlikely for torrent salamanders, but again, for birds, could be, is it just one big breeding population and you can't even really see any structure? And in order to do this, you do different methods. So instead of doing kind of that big range-wide sampling, you want to sample a lot in just one watershed in order to answer these questions. And so to do that, we did that at the H.J. Andrews Experimental Forest this summer. And some of you might know where this story is going. So the H.J. Andrews is a site in the LTR network. Really cool site. It turned 75 this year. It's about an hour east of Eugene along the Mackenzie River. And there are absolute just decades of data here. There's stream data, topographic data, bird data, soil fungi data, and all of it is decades long. And there's a lot of different kind of harvest management and elevation throughout the site. So you can really get at a lot of different questions. And interestingly for us, there was also some Cascade torrent salamander research done in 1998. So we had some known sites where they were and where they weren't. And Hunter did this in 1998, also did a bit of modeling. So we were kind of able to look at sites and say, well, like this has a higher probability, so we'll go in there. and look as opposed to say like a site with a lower probability. And his model did end up being pretty predictive. So our plan was to go to 30 sites, sample 5 to 10 individuals per site, get up to 300 individuals. But then there was a fire, and it's currently ongoing now. It was a bit stressful, but like honestly, like I can't really complain because there's a lot of people that have lost a lot more due to the fire. So I definitely don't want to like take that away from them or kind of minimize their experience. But the fire kicked us all out. It's something that a lot of us are dealing with that work in the forest. It became pretty special to us. But the good thing is that for me, at least, is I was able to sample at 20 sites. So I do have 141 individuals total. So I'll actually still be able to answer a lot of these questions. And a lot of them are going to be really cool. So kind of looking at something like this, these top 5 sites, you can see like 3 of them are within the stream network of all the other sites south of them. But the 2 on the top are on the other side of a ridge. So the streams don't cross at all. So one of the things that's going to be really interesting to look at is, are these things moving over ridges? And that's one of the things that a conservation manager specifically would like to know is, are they moving over ridges or are they only moving within streams? So that stuff is ongoing. DNA is being extracted right now. And so again, yeah, that's kind of what we're doing for conservation. But of course, like there's even something I think maybe more important to conservation. And that is just that, like, people have to care. Like if nobody cares about torrent salamanders, if no one cares about wildlife, then, we can do all the science that we want and it's just going to exist in a vacuum, but there won't be kind of that need to have it used. I mean, ultimately, science is nothing without people. So on the right is Sophia, who was the undergrad working with me in the HJ up until the fire. And, seeing her in the forest was really cool and getting to experience like her seeing her first torrents and really falling in love with them. And, we know how important these kind of personal relationships are in addition to just the science. And so kind of in recognition of that, I, with 4 wonderful humans, did this bilingual children's book called Nuestra Casa with Dr. Garcia, who is my advisor, Isabel Justiniano, who's also in my department and just graduated with her master's in social science, and then Jessica and Jordan, who graduated from undergrad in my department as well. And, we wanted to create this book for a lot of different reasons. One of which was just that, STEM workforce is really underrepresented in the Hispanic population. So 2% of the STEM workforce is Hispanic, but yet almost 20% of the youth is Hispanic. And so there's a big discrepancy there. And then, in addition to just having that, even within the Hispanic community, there's a big difference in the female Hispanics that as opposed to male counterparts and how often they're in the STEM field. And that's something that we kind of wanted to address a bit with this book, just in terms of providing representation. I think like that's what's so, important. So a lot of times, books have been, I'd say, maybe written for a specific audience. And so we wanted to kind of target an audience that maybe hasn't had books written for them. So more kind of the migrant families that are very historically marginalized. And I mean, I'm Latino myself, and I'll tell you, like, most of the books have people with skin color that look like me. So we wanted to specifically kind of target some of the more underrepresented, kind of darker skin income Latino subgroups that exist, to just kind of give those families someone that looks like them within the book itself. And I've actually, doing readings, kind of seen people say it looks like them, and that's really, really cool. And then these populations are also important because they like live at the like outskirt of urban areas. So they're really important at like this kind of rural urban interface. So they interact with wildlife a lot more. They see it a lot more. So it's really something that is, I'd say, more present in their life, and we can have kind of a greater impact in teaching people to live what's in front of them. And then even within the Latino community, there's a big kind of internal bias for working in STEM. I've talked to people in the college that I'm in. Technically, I'm in the College of Ag, even though I'm in Fish and Wildlife at OSU, and talking to some of the people who their job is to increase Latino representation. Talk about how a lot of kids will come and they'll say, I don't want to be in natural resources. My parents said, they told me very specifically, like, I didn't work in the fields to have you work outside. And so there's some of that bias that we need to overcome as well, which again, with representation, we're hoping can help. And then finally, we wanted to do it in English and Spanish, as there's, I grew up in a multilingual household. and a lot of other people have as well. And so this is just something that can make it be shared regardless of the languages that different generations speak. So this is just kind of like some of the pages of the book. And so essentially it follows the story of children who move to kind of a more rural area with their family, kind of discover a we had Portland Metro kind of property in mind when we designed this, but kind of like an urban wildlife area. And then on the kind of bottom, like as the pages progress, we have kind of these diagrams of a Pacific chorus frog, which is the kind of frog character, and then a long-toed salamander. And kind of as the story progresses, you'll see them kind of develop then into these kind of adult amphibians. And the reason we chose those two species is because they're pretty commonly encountered in like gardens and parks. So we wanted to choose a species that could be seen by people. And another part of that we mentioned representation was just having like park rangers and people that could look like the audience that we were hoping to attract. And then having some examples of biologists like Dr. Jenny Robino, who's a mentor of mine, and kind of just again show that that Latinos have a place within STEM, in the STEM workforce. And then we kind of, what children's book wouldn't be complete without some fun information and fun drawings about salamanders. So we have kind of some info about their life cycle and, why they're important, which we try to hammer throughout the story as well. And just kind of finally touch on like the overall kind of arching goal that we had with this was to kind of focus on stewardship and belongings at two different levels. One, just being within the community. I think a whole thing about Nuestra Casa is that like the amphibians have their home there and children have their home in the forest, but they're both part of a larger home, just in their community, but also in nature, and kind of trying to erase maybe some of that separation and have it be more of kind of an inclusive like belonging. And we kind of envisioned it being used as an education tool, being used to bring families together and just generate curiosity and excitement for the environment. So we've paired readings of the book with outdoor activities at places like Tryon Creek or the Winter Wildlife Field Day at the William L. Finley Wildlife Refuge. And then we have done readings at multiple elementary schools as well. And we were able to produce this book with from the Bullet Foundation. And so this book is actually free for educators, and we have a lot of them. So we've done readings before, but if you know of, say, like some schools that would like that and they don't need to be readings, they can just be the books themselves. Feel free to have them kind of go to this website and just fill out the order form. And then, yeah, we can send books out. Our goal is really to see them being used and hopefully being used to kind of stimulate that love for the environment. So with that, just say thank you to my committee members are Tiff, Dee Dee, Lindsay, Tal, and Sarah. And then all of the different organizations that I work with, the Bullet Foundations, who funded the book, and some of the genetic research as well. And then Friends of Tryon Creek, who were very gracious in letting me run. And that's my dog Cheddar. And if you have any questions, you can just email me at this email. And hopefully you enjoyed the presentation.
Yay, thank you so much for this really enlightening talk. Folks can ask questions in the chat. I will start with my questions that I have, and as others can filter in some of their questions. But I'm curious, I have some very easy questions. What made you, I have some ideas, but what made you interested in torrent salamanders specifically?
It's actually kind of funny. Yeah. So when I was starting my PhD, I just had a job kind of leading that eDNA stuff. And I initially didn't think it would be part of a dissertation, but I just really liked it. I've always really enjoyed finding salamanders. It just feels like such like a mysterious animal, like they're always there, but yet you don't see them. And there's something really attractive about that. But having studied them so much, they're also just really cute. Like they look like glass frogs, I worked with them for 4 years and still every time I find them, I'm like, oh my God, yes, here they are, So I just really like it for lots of reasons, I guess.
Cool. And Little would ask if you find tailed frogs as well.
Yeah, so yeah, we find tailed frogs often. They're kind of a good indicator that we're in the right spot to find torrents because they also use a lot of that headwater. But what's kind of interesting is I think tailed frog tadpoles can kind of have a little different habitat requirements. So say in like the H.J. Andrews, we actually almost never found tadpoles, but then we were in say the Coast Range, it was very so there's a lot of overlap in habitat, but maybe not like a perfect overlap, I guess.
Cool. Yeah, a follow up to that. If we wanted to see torrent salamanders on ourselves, where should we go and
Oh,
when should we go?
well, I can actually answer that. Like I said, Columbia River Gorge is by far, I think, the easiest place to find Cascade torrents. And if you're on the Coast Range, then kind of in the streams around Tillamook up in the hills. Again, they like more of that higher elevation stuff. Um, but I would say in the Columbia River Gorge, I'll let you know, like, Starvation Creek is a very popular hiking area. You just kind of look under some rocks around the little seeps that run into Starvation Creeks. You'll probably find one, but just be careful when you're like lifting and then like never try to put like a rock back on them. Like try to put the rock down and then like they'll go under it.
That's a great tip.
Yeah.
Um, Willow asks, what is the primary food source of a torrent salamander?
Yeah, torrents eat macroinverts. So when you hear people say macroinvert, I just am saying this because I learned it the other day and I didn't know for like years, but it just means that you can see it with your eye. Anyway, so they eat a lot of macroinverts. Like stuff like springtails, really whatever is in the stream. So just invertebrates. They're uh carnivores, even though they don't look like them.
Yeah, they're just too cute. Uh, and also, what plants do cascade torrents depend on being present in their habitat? Are there any, yeah, plants that they're dependent on or anything like that?
Yeah, that's a good question. And yes, but I don't think it's like for maybe the direct reason that a lot of people think. I mean, plants are really important in these headwater streams because they tie the soil together, and that's one of the big things that we can see happen after fires, right? Is when a lot of that kind of the ferns and uh like the vine maple and those kind of plants that really salix species that really have like strong root systems and kind of hold all the soil together. When we see those be lost in burn sites, what we end up seeing is just like a big input of sediment that again kind of fills that like space that's needed between gravel. So it's really that stream side vegetation that's really important to them.
Cool. Another question is, are salamanders damaged by handling with their hands, or anything like that?
Yeah, that's another good question. I always use gloves when I handle them. Every salamander is gonna be a little different. But you know, I feel like the thing that I'm often try to be most cautious of, especially with torrent salamanders, is one, drying them out, and then two, raising their body temperature. And so a lot of the oils on our hands can kind of do that. They can remove the moisture, and all these salamanders, especially the ones in the water, have this kind of protective mucus. And so the more you would handle them with bare hands, the more that mucus is being removed, and then that can actually kind of reduce their ability to thermoregulate and take in things. So I use gloves when I handle them in the field, and then even while using gloves, I'm pretty much like, sometimes we're doing tail tips, some are a little more squirmy than the other. So I'm like constantly putting my hands either back in the water to kind of cool them down a little bit or just to get more moisture on them as well.
Yeah, that's really helpful because I personally I have a nephew that loves catching frogs
Yeah,
since he was like a toddler. He's like a toad whisperer.
yeah.
But now I can have that in my brain for him
I
when
mean,
he's maybe handling salamanders.
I would say, it you still can, but like, you put your hands in the water, you get them cold, you make sure that they're always wet. If I'm handling stuff, again, I try not to, but sometimes to take photos, like more of the terrestrial stuff, like the Del Norte I showed, just rubbing your hands with dirt to like get rid of that oil and stuff can be helpful. So just, and just minimizing time, really. But
Sure. Yeah,
often you can just, use them.
that makes sense. Yeah. Um, is there any similar research being done on the Olympic torrent salamander?
Not as far as I know. We actually collected some samples of the Olympic torrent. Like you saw kind of in that PCA plot, we saw that really divergent population. And one of the things that we're trying to do now with that is just kind of understand how related it is to other populations. So we're doing like a little bit of that, but not really. I think there is a researcher, I forget, I want to say her name is Lauren Chan, and she's doing some of that work with Olympic torrents. Um, but I'm not sure about the eDNA side of it. I know that the Washington Department of Fish and Wildlife is actively working with Olympic torrents.
Cool. And then Anne asked, does that fungus disease that has killed so many tropical frogs occur here?
Yeah, chytrid. And it actually does, yes. We know that it's here. It hasn't resulted in those mass die-offs. It actually doesn't impact salamanders. There's actually another one for salamanders called B-sal. And that so far is not in the United States, but it's a big concern, so much so that I know that researchers at Washington State University in Portland are working on it right now, where they're taking a lot of different species and subjecting them to B-sal and trying to understand just how many of them are going to get wiped out. Because yeah, like that species, that chytrid that's being talked about, when it's in certain environments that again, like maybe have lower genetic diversity or just populations that haven't been adapted to this, it can just completely wipe out populations. And so, but it doesn't seem to have that effect here as much.
Great. And just two more questions in the chat right now. The first one I'll give you is, can you expand on the specific roles torrent salamanders play in the ecosystem?
Yes, I sure can. So there, in a lot of these headwater streams that again don't have fish, they and Pacific giants are kind of the dominant vertebrate predators. And so it's not really known if they're exerting top-down control, so like their predation is controlling the number of invertebrates, or if it's kind of a bottom-up where it's just the number of invertebrates means there's more or less torrents. We're not really sure about that. But that's an important role. And I think also just as prey items. So we talked about how tied these streams are to the upland habitat. So a good friend of mine, Marie Tosa, is a huge skunk biologist. Well, I mean, she's really just an amazing biologist, but she really likes skunks, and she's done some work in the HJ with eDNA, but on kind of stomach contents and feces to try to understand what they're eating. And torrent salamanders are popping up. So we know that skunks eat torrent salamanders, and it's likely that some other species do as well. So I think they're kind of important for linking that kind of terrestrial and aquatic habitat, both as a consumer and then also as prey.
Yeah, just a follow up on that. Can you give examples of the common macroinvertebrates that they eat?
Yes, but it's a very rough one. So like, stoneflies, dragonfly larvae, sometimes we do find them sometimes in their stream. But I am not a huge like a macroinvert people. So when I see them, I'm just like, oh, that's a cool one. Like that's like a shredder or a grazer, but I don't know what kind. But a lot of it's really that larval for the stuff that then flies out, like stoneflies or stuff like that.
Wonderful. Yeah, that's helpful anyway. More than I know at least. And then last question, at least right at this moment, is did you run across any data on the large mountain salamander?
Yeah, it's funny that you ask because I also have another project because being a PhD student means having 30 projects at the same time. That's on the large mountain salamander. So actually with that, my mentor Dee Olson, we just wrapped up our third year this year of doing sampling for large mountain salamanders. We kind of matched burned sites and then unburned sites to understand how they're responding to fire. And so, yeah, I'll actually be analyzing that data this winter. The one thing I will say is that over the years since the fire, we have found them more, but they're also another species that can be really difficult to find. And we've had sites that we've sampled, twice a year for 3 years, and we found them one time. And so we know that they're there, but those are species that are they're talus specialists, you know, so they live in those talus slopes, where, you know, as you can imagine, there's a huge amount of 3D structure. Talk about underground. I mean, they go down way down. And so we think that they're able to withstand fire pretty well. Because there's a pika researcher that I know that is looking at pika at the gorge, and they have temperature sensors down in the talus when the fire came through, and they saw like negligible increases in temperature. So they're really able to just kind of use those as refugia and ride them out.
Oh my gosh, that's fascinating. I can't wait until you give us a talk about that next year when you look at that data.
I need more than that. Yeah.
Um, yeah. What is the range of the large mountain salamander? Are they in Bull Run?
Yeah, they are in Bull Run. We're the Bull Run is, I think it's that watershed that's the drinking water one, no? Like that one's actually off limits even to researchers. But we have some right adjacent to it. Large mountain salamanders are actually a really cool species. Again, I mean, if y'all want to see those, the Starvation Creek is actually a fantastic spot. This is kind of a, maybe you might not want to do this unless you're in a group, but if you go when it's raining in the spring at night with headlamps, they will blanket that concrete trail that runs along the highway. And they almost look like little fish because there's so much water on there that they look like they're swimming. But yeah, so those guys are found on the Columbia River Gorge, both on the Oregon and Washington side. But then they also have an interesting, like, disjunct population by Mount Rainier. And I know because, like, when, you know, with climate change, it's getting harder and harder to time surveys for large mountain salamanders because oftentimes if the temperatures are below freezing, the detection probability is almost zero. But then if it's dry, then it's also zero. And so that window of kind of overnight freezing temperature stopping and then the rains continuing has gone a lot more narrow, especially over the last 3 years. But we'll have the people from Rainier be like, well, they're active right now. I'm like, that's cool, but like it's 25 degrees here.